Okay, so thank you for joining me on this recording. And as I'm sure that many of you have figured out at this point, so we're working on week 13, but we're looking at the chapter of the fluid, electrolyte, and pH balance because of the outage on the Maricopa system um, online resources and because of the extension of the spring break for 1 week due to that. So we're running 1 week behind, but we'll make it work. So what I really wanted to do this week was to start by going through the feedback from the exam 2 and practical 2. So some of the common things that came up in your answers, all of those have been finalized. The grades are up there. And then I wanted to mention a couple of words about the exam 3 and practical 3, which are coming next week. But majority of this session, as always, I want to spend focusing on this week's module. So go through the assignments, the due dates, the scores, and there's a new type of assignment that we'll be doing on this week's module, which is the critical thinking assignment. So I want to take a little bit of time on that to go through that. How does that work? And of course, we then go through the main points of the chapter material. I will probably skip the part about the expired Connect access code. I have a feeling that it doesn't affect this class as such. That's something that if it does expire and you have used it, for example, from the previous semester, reach out to me and we'll see what we can do. It's usually through the publisher that we would look at that. And then just kind of the general things that we tend to speak on a weekly basis, what to do if you're missing or want to retake any assignments, tutoring options available that I'm happy to help with those and any questions that you may have. So without further ado, let's jump right to it. But before we get too much further, I do want to apologize if there's a lot of noise on the background. I'm actually recording this from my own home, and I did get recently young baby chicks, and they're staying with me here inside the house in a cage until they're a little bigger, and I know that they'll be fine outdoors in their own coop. So for that reason, you might hear them making noises on the background. We'll try to make it work, but if that gets too crazy, then we'll have a look of what can be done. But they seem to be getting ready to go and have a nap. In terms of the last exam and practical, so those were the second one of each, I thought that maybe we could start with the practical and have a look of just of the general themes first of all that came up. So I really encourage that whenever you're a little bit lost and you don't necessarily have the full answer, try to give something that's kind of a close answer at least, or tell me what you see on that picture, other structures that you can identify. Give me something that I can do to award at least partial points. I normally try to give at least some points if you have tried something. So give me something to work with and I'll try to work with you as well. Really, the biggest themes that came out as a kind of a whole class, I just want to remind you of the basics for the chapter on the heart. One thing that I want to make sure that we're all familiar and remember the chambers of the heart. So we have right atrium and right ventricle, left atrium and left ventricle. And it's important that you have the basics correctly so you can tackle some of the little more challenging questions. So I'm just going to make a really quick sketch so we remember we could draw heart as simply as a box, and The right side is, of course, going to be the patient's right side, so not our right side. And we find both on the left and right side the atria at the top, so the more superior chambers are atria, and then the ventricle at the bottom. So they are more inferior chambers. Another thing that I think is worth noticing is that we have certain major vessels that relate to the heart. So we had our inferior and superior vena cava coming to the right atrium. So of course, superior vena cava covers, drains the head, neck, and upper limbs, whereas inferior vena cava drains the rest of the body. We will also have a pulmonary trunk coming out from the right ventricle, so that goes to the lungs. And then we had our aorta coming out of the left ventricle. And that, of course, was what supplied the blood to the systemic circulation. In terms of the coronary arteries, these were the arteries that were the first ones to appear after the aortic valve. So I've gone through that. There's 3 that I want you to know. So the 2 that appear immediately after the aortic valve are going to be the right coronary artery and the left coronary artery. And only after we continue the aorta up and get to the aortic arch, then we get the other branches such as brachiocephalic and so on. So it's the coronary arteries that are the first branches. In terms of the heart and coronary supply there, let's just sketch really quickly a little heart here. So this would be our atrias and our ventricles with our apex pointing to the left, downwards, and to the front. We can see our superior vena cava coming in and inferior vena cava. What else could we see? We could see our pulmonary trunk dividing to right and left, and we can see our aorta, which is gonna loop here and continue to the rest of the body. So there's a pretty good sketch of the heart. We're gonna focus on the actual heart muscle on this discussion of the coronary arteries. So this is looking from the front as if you were taking the chest away. And let's just add our coronary arteries to that. And we're going to start with the right coronary artery, which is going to be then running here on the right side of the body. And then we had our left coronary artery, which divided almost immediately to circumflex artery, which is going to loop around and then go to the back of the body. And then we have our anterior interventricular artery, so runs on the anterior interventricular groove, also sometimes known as LAD. And it is the anterior interventricular artery, or LAD, if you wish, that supplies the anterior side of the heart, and it is then the coronary artery, right coronary artery, and the circumflex artery that supply mainly the posterior side of the back side of the heart. In terms of the histology, I really encourage you to try to be as specific as you can. A lot of the time, partial points are available, but if you are super specific, you can actually get extra points on those. So just try to give as much detail as you can. And histology is covered in the practical exams just as gross anatomy. So I know that it's often for some people, including myself, easier to think of these themes when they are structures and organs that you can see with naked eye. But histology is an important part of the anatomy and physiology, so we need to have a basic appreciation of that as well. So let's have a look at certain particular questions that kind of draw my attention in terms of that I want to just make sure that we're all clear of those. So I wanted to remind you that sometimes what happens is that we have structures where we actually have multiple names that would be possible when we discuss them. So here, for example, let's just orient ourselves. So what we are seeing a heart where we have now our right atria here. So this would be our right ventricle, and we would have our left atria here and left ventricle here. And we're being asked about this particular valve that we see here. Well, because we see that there's a pulmonary trunk coming out of that valve, we know that that's going to be going to the pulmonary circulation. And we, of course, see that it's coming out of the right ventricle. So there's no other option than it being the pulmonary valve. So that's the name we've used a lot. But another name that we could see being used about it would be semilunar valve. So both of those would be correct. If you used one or another, you got full marks. But if you used both, then you could get extra credit for that. So I really encourage you to provide me with all the information that you can come up with, and I'll definitely try to award that whenever possible. Another example of that we see here, kind of a heart located very similarly. Let's have a look here. We have our right atria, right ventricle, left atria, and left ventricle. So this valve here is the one that separates left atria and ventricle from each other. So we might have heard different names for those. One was left AV valve, which stands for, of course, the atrioventricular valve. The other name that you might have heard was bicuspid valve. So that again describes the number of cusps, those leaflets on this valve. So 2 of those on this case. Or then the name mitral valve, which is one that's used as well. So there we have 3 different names. And again, if you gave me just one of those, you got full marks. But if you gave any more of those, there was a chance for those extra credit points that you could gain. So I always encourage you to have a look if that's something that you can do. And I know that at the background, my chicks are getting a little bit more awake, so they're making a little bit of noise. Don't let that throw you off. The other thing that I wanted to mention in terms of the practical, that I realized that sometimes there's just a lot of material to know, but try to be as detailed in terms of when you're studying those structures and you're naming those structures as you possibly can. So let's have a look of what we could see here. So we see, of course, our right atria that has been opened on both pictures. And here we see then the right ventricle, left atria would be here. So we see that there's a structure within the right atria that has been circled. So what I was looking here was especially this structure, which can be seen on the wall of an adult heart as well. But this structure is something that I was really looking. If you gave me an answer that it is right ventricle, you might have gotten small amount of points, but that was not really what I was looking. What I was looking was a structure known as fossa ovalis. Fossa ovalis is a structure that plays role mainly early development of your life. So again, let's draw our heart here. And I'm going to use the simple version. So our right atria, right ventricle, left atria, and left ventricle. So we remembered that the pulmonary trunk from the right ventricle went to the lungs. And the aorta from the left ventricle then supplied the systemic circulation, rest of the body, all the tissues there. Well, before you are born, before the birth, you are in the mother's womb, so there's really nothing that your lungs can do in terms of breathing. You can practice those processes, but there's no air to breathe. So it really doesn't make much of a sense for our body in a baby that hasn't been born yet to circulate the blood to the pulmonary circulation. So for that reason, there's been special adaptations that have developed. And one of these was this fossa ovalis. So what fossa ovalis does is that, when I've been to hide that, is that it allows blood to bypass the lungs and make its way straight from the right atria to the left atria. So completely crossing out this pulmonary circulation. And this would of course make sense since we don't have functional lungs. Why to waste the resources of pumping the blood into there? They become functional the moment you are born. When the baby starts crying, that's a sign that things are going well. Baby is breathing. Baby has taken its first breaths. And we all know how important it is to secure the constant oxygen supply to the brain and rest of the body. So if that doesn't happen early on, that's a huge problem. So when we start breathing, right when you are born, actually this fossa ovalis structure closes. And what happens? It completely closes, but we can still see the remains of that in an adult heart. So that was what I was looking here on these pictures, that you would identify that structure. In terms of the spelling, I try to emphasize that never worry about the spelling. I do not deduct points of that. I go always manually back and read every answer that you've given several times, at least 3 times, blinded to whoever gave those. So I really do want to make sure that you don't worry about that spelling. I will go later on, even if the machine automatically grades you to lose out on some of the points, as long as it's resembling something that I was looking for, it makes sense, you will get the full marks. There is no grade deduction whatsoever. On this diagram, I just want to show, just because this was something that came up a few times. So I got few answers that what we see here is, let's start by orienting ourselves again. So we see our left ventricle and our left atria. So here, left atria and left ventricle. And I got few suggestions that what's colored on this blue area would be left ventricle. Well, if it was left ventricle, I think we would have colored it a little bit larger. And what we're showing there is actually the wall lining of the left ventricle. The other answer that I got a few times was that maybe it's a papillary muscle, where papillary muscles connect the chorda tendineae of the leaflet, the valve of the heart, to the wall of the ventricle. So they're kind of continuation of the chorda tendineae. So that wasn't what we were looking. What we were actually looking was trapecular carne. And what trapecular carne does is that it actually prevents blood from pooling to the ventricular area. And why this is important is that By preventing the blood from pooling, we're preventing that the blood would start clotting in the heart. And if that was a clot, that would be, of course, a major problem. We don't want a clot traveling to anywhere into coronary circulation or pulmonary circulation or really anywhere else in the body. So by having this structure, this adaptation of the surface of the ventricles, the trabeculae coronae, that prevents the blood from staying behind there and kind of really makes sure that all of it gets squeezed out and all of it gets mixed up. So no blood just remains in one place and start to clot. Again, no great deduction if you had issues with spelling. This was another one that's a kind of a tricky one to spell, our tonsillar crypt. So again, don't worry if you initially don't get the points that you think that you should have gone. I will always go back and manually review those. If you provide me some extra details, you have a good chance of getting, again, extra credit. And here, for example, I was looking for you to identify that what we're seeing here is the pulmonary artery. Remember that pulmonary system is a little different from rest of the body because in pulmonary system, the arteries carry deoxygenated blood and veins carry oxygenated blood. And that's, of course, the reverse in elsewhere in the body. But if you picked up the pulmonary artery, you got full points. If you also told me that what we're seeing here is the left side of it, because we're here looking at the heart from behind, from the posterior view, so patient's left, you could get extra credit for that. So again, if you are comfortable with what you're seeing, try to provide me some extra information. You might gain those extra points. One thing that's kind of already something that I think we should have covered when we covered, for example, the bones, or when you talked about the nervous system, the difference between radial and ulnar side. So we remember that radius is on the thumb side. So one of the mnemonics that one of my students told me that they use is that they think of, you put your thumb up when you're happy and you say right. That's correct. So you say right on for radius. So radius is on the thumb side and ulna is then on the pinky side. And this goes back to the theme that we've seen before, that it's really worthwhile to learn the basics, especially in anatomy and physiology, because you can kind of reuse those time after time. So if you learn the bones correctly, then it's super easy to know the nerves, then it's super easy to know the blood vessels, and so on. So we would be able to separate the radius and ulna and the radial artery and ulnar artery and radial vein and ulnar vein based on those. So that's something to notice there. Again, I do give more points, even though it's often when we talk about structures and in anatomy and physiology, we start abbreviating things. We start leaving things out because we speak and we cover those so much. But in an exam situation, try to give as complete answer as you physically can. So for example, here, looking at this artery that we see here, I would easily say, yeah, it's the ulnar. But remember, if you can, to add that it's an ulnar artery when you're giving the exam answer. So typically on a practical, I try to reward that. You might get extra credit, but I won't give the extra credit for every time you give a complete answer. First time I might give a little more, then when you give a second time, a little less, but still some extra credit. And eventually I don't give extra credit for every time because you really should be using the complete answer. But if you don't use the complete answer, you miss out on those little extra credit points that we have available there. And of course, you see now pinky side, so that's going to be the ulnar, just as we just discussed. Sometimes things are named in anatomy and physiology after other structures. So here, for example, we see subclavian vein. So subclavian vein here runs under the clavicle, so below the collarbone. So try to learn those if you knew the bones. That then guides you on identifying other structures that refer to those bones, such as clavicle. So try to make sure that you connect these. Systems are not separate ones, and the names often are linked as well within the same area. If you are talking about vessels in particular, the nomenclature is very important in terms of being very specific about the locations, because the locations tend to change in terms of the blood vessels quite a lot of times and quite quickly. So we'll see the portion here running right underneath the clavicle was subclavian, but then closer to the heart, it turns to brachiocephalic. So make sure that you're kind of seeing where those transition points take place, and you know as many of those as you can, so you're not just guessing that, oh, one name is going to cover the entire blood vessel. Blood vessels are, unfortunately, same as nerves, one of those things that you just kind of have to learn that they change names as they proceed from different parts of the body, even though it remains as the same larger vessel in an anatomical and physiological sense. In terms of the things that are further important, we want to be sure about the location. So let's continue having a look of that. What we see here is a common carotid artery. So common carotid artery covers the part that runs on the neck. And again, if you wanted to get more points, you could have told me the side. So on this diagram, of course, it's going to be left. And common carotid artery then divides into external and internal carotid arteries after the neck. So just be sure that, again, you remember what location, what part of that vessel are we looking at. Here's some other examples of locations. So I got 2 times students telling me that this structure here was going to be common carotid artery. Of course, we remember that common carotid artery covered the neck region, so it would not be that. What we were seeing there instead was the brachiocephalic artery. And I think that was shown nicely on this one. So again, sometimes it requires looking both models, both diagrams, and both plastinated models or pictures of plastinated models. So try to look at those and combine those, look all the sources that you have available. Sometimes we might be, again, using the same term for many different systems within that same region. So, for example, here we see axillary artery, and here we see axillary lymph nodes. So we remember axillary referring to the armpit region. So if you learned the axilla as a term to start with to describe this region, then you can use it in so many systems. It's definitely worth of kind of putting in the effort at that initial point to learn the terminology, because it's going to make it easier to learn the rest of the term. It's also something, if I'm totally honest, I remember when I was a student, it felt like a lot. The more you do, easier it becomes, and really the anatomy and physiology as a language becomes more familiar and easier for you to handle, more you cover that. Then what I have here is that if we have same organs, say for example here we were talking about tonsils, so we have many different tonsils. So then if you're able to specify the location of the particular tonsil, such as here the pharyngeal tonsil, that of course would gain more points. I think that you could get double points in that if you included both, than rather just saying that this is pharyngeal region or just saying that this is the tonsil. So try to make sure, again, when you're learning that, oh, you see that, oh, there's many tonsils, maybe I should know what's the difference between these different tonsils. Let's have a look of the next one. If you're able to provide some extra detail, that's always good. So what we see here is a dissection picture of an adult, and we see that there's the remains of what used to be thymus. It has been replaced by the connective tissue and adipose tissue. So as you age, your thymus gets replaced by those, but it is still there. It just becomes harder to see. So if rather than just saying thymus, which was full marks, completely correct answer, but you told that it's the thymus that we're seeing there, what's left, and kind of referred to this concept of thymus being replaced over the age. You could get extra points for that as well. So the other one that I do want to make sure that we're just really familiar, this in general wasn't a huge challenge, but I just want to ensure that we're all on the same page again. So make sure when we had question where I was asking you to tell apart the pulmonary and aortic valves, make sure that you remember which one comes from which chamber, which ventricle. So again, as a reminder, here we see the right atria, right ventricle. And from the right ventricle, we have, of course, our pulmonary trunk coming out, and pulmonary system led to the lungs. Whereas from here we have left atria and left ventricle. From the left ventricle, we would have our aortic valve, and that would then give rise to the aorta, and then to the systemic circulation to all parts of the body other than the pulmonary system. So again, if you were able to make sure that you remember the difference between those two, that would be super helpful. So what I've done here is actually labeled everything that we just talked, just rather than just labeling the chambers, I've taken care of the label also the valves. So you remember that right atria and ventricle are separated by right AV valve, also sometimes called tricuspid valve, because there's 3 of those cusps, leaflets there. And from the right ventricle to the lungs, we go through pulmonary valve. So this would be pulmonary trunk right behind it. And what you'll also notice, what's kind of a worthwhile to notice, is that it is this right side is deoxygenated blood, so blood with low oxygen concentration. Then let's have a look of the left side. So left atrium to left ventricle, blood has to go through left AV valve, which was sometimes known as bicuspid valve, so 2 cusps, 2 leaflets on that valve, or mitral valve. And from the left ventricle to the aorta, we go through aortic valve. So that was the important one. It's not really showing up great on this picture, but it would be hidden a little bit behind what we see there, the pulmonary trunk starting. Remember, on the left-hand side, we have oxygenated blood, so the oxygen concentration is high here. So that's all I wanted to say about the practical. I also collected 2 points to note about the exam. So let's go through those quickly as well and have a look together. So you've noticed that not in all exams, but often in exams, I try to include an extra credit question. And extra credit questions are what the name suggests. That's your chance to get those points. If you missed out on something at the exam, you can still get those points back by tackling that. Usually I do mention the extra credit question largely what the theme or topic might be. I don't always have the extra credit question. So for that reason, if you're interested in tackling those, I do recommend those revision sessions before the exam. But it's worth trying that even if you wouldn't know perfect answer, just so you get some of those points from there. And then in terms of the different chapters that we covered in the exam, we talked about the heart. So that brings us to the theme that we just talked about. Know the difference between right and left, and that the right side of the heart carried the deoxygenated blood. So this blood is low in oxygen concentration. Whereas then the left-hand side, now coming back from the lungs, it's going to carry oxygenated blood. So the oxygen concentration is high on this side of the heart. So just make sure that you're familiar and comfortable with this concept. If we want to draw the heart here, remember again our right atria, right ventricle, left atria, left ventricle. It is going to be that to the right atria, we collect the blood from the body through superior vena cava and inferior vena cava. And this blood is going to be deoxygenated. So when it heads to the pulmonary trunk and then to the lungs, that's going to have low oxygen concentration as we saw there. But when it comes back from the lungs, it's of course going to have high oxygen concentration coming to the left atrium, then left ventricle, and from the left ventricle to the aorta, and then to the rest of the body. So we have now a lot of oxygen. That's why we take it to the around the body. Other thing that I asked, which I think was a little bit of a challenging question, but a question that I had mentioned and warned you a few different times, was that the coronary arteries were an important one to know. And coronary arteries are the first vessels immediately after the aortic valve. So many of you were thinking of these branches from the aortic arch, such as brachiocephalic, which is the first one. But I wasn't asking of those. I was asking the first vessels immediately after the aortic valve, and those are of course going to be our right and left coronary artery. And the left one then divided further to the anterior interventricular artery, or left anterior descending, which is the other name, and then the circumflex. The other thing to know about the heart, if we talk about general, when we have patterns of some kind, such as order of events like an electrical conduction of the electrical impulses through the heart, do study the order of events so you can tell how things spread through the heart, as we discussed, for example, on this question about the electrical conduction. Also, take the time to think, not just memorize. Why would we have certain things happening? So we saw, for example, that at the heart we had slower action potential than in the nervous system. Why would we have slower action potential propagation than in the nervous system? Well, that's of course for the purposes of what the heart is all about. And what the heart is all about is of course pumping that blood from one chamber to another. So it's all about for the time for the blood to travel from one chamber to another. So we give it that time. So that's why our action potentials aren't rushing through different parts of the heart quite as fast. In terms of the questions that I had about the different blood vessels, I want to make sure that we remember the differences between arteries and veins, what their functions are. You remember that arteries were directly from the heart, so they were coming away from the heart. So here's our heart. So what they do, they have to deal with the pressure fluctuation they have to deal with high pressure, they have to deal with the pulsatile flow of the blood. So they are often very elastic. They do the job of kind of being able to handle that that pulsatile function and handle also the high pressure. So arteries tend to have thicker wall than veins, which brought the blood back into the heart. So veins instead have thin walls because we want to have that venous return concept, that veins get easily squashed by the skeletal muscles. And as veins get squashed by skeletal muscles, those squashing helps to push the blood back towards the heart. But what happens also, the backflow, when there's no squashing, that's prevented by having valves in the veins. So we don't have that backflow. So that's one important adaptation that we see within the veins. With the vessels, we see that sometimes we have multiple names for different or for the same structure. And if there's multiple names, I could really ask any of those. So just be certain that even though you might prefer one, you have at the back of your mind the understanding that it might have other names. And finally, I want to remind you, when I tell you mnemonics, it's usually trying to lead to something. It's usually giving you a simpler way to tackle something. So for example, here we were talking about the signs and symptoms of a shock with the mnemonic of PC fats. That stands for police constable, that's fat. So that's a very British mnemonic. So that told us the signs and symptoms of the shock. And we'll have a look of that in a few moments' time. In terms of the lymphatic system, probably one of the questions that I really emphasized multiple times was that when does the interstitial fluid become lymph? Because it's the same composition. It was when it entered the lymphatic system, those lymphatic vessels. So again, if something's being mentioned a few times on the lectures, that might be a good one to remember. So that's why I do recommend watching the lecture videos and watching the revision videos. And of course, one big one that I think that is a fair one that got asked, and most of you did good on that, but some might just want to double check that. We have multiple functions for the lymphatic system. So first of all, of course, we know the pathogens and dealing with invaders of the body as an important function of the lymphatic system. But other functions involve regulating the fluid balance, so moving the fluids from one part of the body to another as needed, and also the passageway for the dietary fats that were too large to be taken to the cardiovascular system, capillaries of the cardiovascular system from the digestive system. So they get actually dumped into the lymphatic system that then transports them close to the heart and there dumps them to the blood. So those were some general points to note. Now let's look at particular questions and what should we kind of pay attention or have at the back of our mind when we're working through. One of the harder questions in my mind are the questions where we're asking which option is not correct, because if it is a question where we're asking that what is not correct, we're left with more answers that are correct than that are incorrect. So the best way that I can think of is to eliminate the correct answers, and then you're left with the wrong one. Here, for example, we were talking about apex pointing, but we remember that apex actually points inferiorly, so downwards, not superiorly, and it points to also left, not to the right, and it does point also forward, so anteriorly. So there was just an error on the direction where the apex pointed, but that made it not correct. So make sure that you pay attention to even the smaller details on these questions where we're asking what is not correct. There's more correct answers than incorrect ones. Sometimes it's easy to jump that, oh, I know this is correct, and then you don't read the keyword, which was the not here. I had another one, rather than saying not, it was just hidden there as an incorrect one, statements regarding heart valves. So it might be that you were, for example, looking at the question quickly and you were like, oh, that's true about heart valves, that heart valves facilitate the unidirectional flow of blood. That's correct. And, or that, yeah, heart valves, the opening and closing of them is regulated by the pressure changes, or narrowed heart valve can cause the rest of the heart having to contract more forcefully. Hey, that sounds correct. But make sure that you do not answer that based on what's correct. You answer what's incorrect. Here, another correct one would be that failed valve can result in backflow of the blood. But what I was looking here was, again, a slightest of detail that changed the sentence incorrect. So here, for example, I talked about the faulty heart valves and using mechanical valves with them. Well, we don't tend to use mechanical valves in younger patients who are still growing, because mechanical valves, of course, cannot grow as the heart of the young patient grows. So either we would have to do another surgery or multiple other surgeries, or that valve would be wrong size in comparison to the size of the heart. So we try not to use the mechanical valves on the younger. So that little detail made it incorrect in this case, as we saw the question. Sometimes you have questions where I'm asking you to check all that apply. So there might be multiple answers that are correct. So some structures, you remember, for example, had multiple names. So for example, here, I was looking for the supply of the anterior portion of the heart, which is of course by the anterior interventricular artery. But in addition, the other name that I thought you about that was the left anterior descending. They're the same thing. So ideally, for full marks, you would have chosen both of them. But if you only chose one, you got partial credit regardless. Here's another one where all that apply. I was looking for the sources of the valves that make the second dot sound when you listen with stethoscope. So of course it's going to be the aortic and pulmonary valves, but you remember that together they were known as semilunar valves. So again, there was more answer options hidden there. So try to get all of those, otherwise you'll just get the partial credit. What else did I have here? The one that we already talked about, the first vessel after the aortic valve, it was, of course, the coronary arteries. Many of you went and jumped right to that aortic arch, which is the larger structures, easier structures to think of. And of course, on the aortic arch, then, the first one is the brachiocephalic. But that's not really what we were looking there. This is not to the scale. That's our heart there. So right after the aortic valve, we had our coronary arteries coming out. So that's what I was looking there. And it was mentioned in the lecture. It was mentioned in the review session as well. So it's about paying attention also to those more little facts. I mentioned that it is word of learning the order of events. That can be type of question that comes up. So for example, here I was asking about the order of events of electrical conduction from sinoatrial node to from sinoatrial node to covering the atria, then reaching the AV node, where there's a little bit of a delay or relay station, and then moving to the bundle branches, Purkinje fibers, and ventricles contracting. So that was the right order. I think we covered that with quite a nice animation. So if you are a visual person, you might want to think of, when you see those animations, where do things go, and formulate your memory of that based on that. Or simply, if you prefer, just memorize the step by step what happens, the order of events. Those make very common and very good questions in the exams. Again, if you can connect it to material that you already know, that's super useful. So we have seen the action potential in nervous system already before. And what we saw with the action potential in nervous system was something like that. Here's the resting potential. And then we depolarize, shoot up. We might go over 0 millivolts a little bit. Then we have the plateau. And then we depolarize. We do a little, or repolarize, sorry. We do a little undershoot, which is the recovery time. And then we go to the normal action potential, which was negative 80 or 60 millivolts, depending on where we are. So here we already know some of the terms, and when you look at the cardiac action potential, you see that it doesn't have all of the same elements, but very similar elements. So although on the cardiac action potential, there's a slight downward slope, it is still our plateau, the area where we're staying almost flat here. So use the guide of what you already know to assist you when you're trying to learn structures or learn concepts. Sometimes we do have multiple names of the same structure. So we talked, for example, here that semilunar valves can be also known as the pulmonary and aortic valves. So learn all of those names. Sometimes we have diagrams. I can ask also about those in an exam. So here we had a nice diagram showing different kinds of capillaries. And what we saw, I had mixed up the order a little bit. Let's start with the first one. First one is here, our fenestrated capillaries. They have small windows on the side. So that comes from a French word for a window. Then we had another type, which was a continuous capillary. So there's no gaps whatsoever between the cells that form the lining of that. And finally, the sinusoidal capillaries, which have those big openings. Sinus is a big opening of something. So I was asking you to identify those on this picture. So that could be something that I do ask. And then kind of utilizing that information, as we saw here with the sinusoidal capillaries, those have the largest gaps missing. So when there's large gaps missing of the wall, the speed slows down of the blood that travels through there, which then allows to do the exchange better than in any other type of capillary. So that it goes a little bit on the side of the physics, but very little. So again, there is a connection between form and function to know this. Sometimes it helps if you don't know a question in an exam to look, does it come up somewhere else? And that happens quite a lot of the time. So something else can remind you of that. So make a note, physically or mental note, to come back to that and have a look if there's something that might help you to remember that later on. Using mnemonics is a very good way, especially in anatomy and physiology, where you have a lot of things to remember. So, for example, here I was asking for signs and symptoms of a shock. And for that, we had the mnemonic PC-PATS. So P stood for pale, C for cold and clammy, F for fast and weak pulse, or fast and shallow breathing. A for that these patients are often agitated. D, they might be thirsty. And S, that they're about to head to the shock. So again, when you have a big list of things to identify, it does help when you have a mnemonic so you know exactly those terms that you're going for. So I recommend using those as much as possible and making your own ones as well. Whatever helps you to remember things, go for it. Sometimes we have definitions. This particular one I mentioned multiple times that I'm going to ask it because it's a one that comes up, that when does the interstitial fluid become lymph? Well, it became lymph at the moment when it entered the lymphatic vessel. So that was just simply a definition memorization. The contents of the interstitial fluid and lymph are exactly the same. The difference comes where they are in the systems. So that's all that I have to say about the review of the practical 2 and exam 2. And that brings us to talk next about next week, where we are going to be seeing, and I'm going to have to change that week. That week is, of course, going to be week 14 now with the new schedule. So exam 3 and practical 3 are available all week. There's no new materials that we cover next week. I also offer the review session, which focuses on the exam material on both Monday and Wednesday, both in the morning and in the evening. So at 10 o'clock and in the morning and 8 o'clock in the evening. So I offer both of those. On both Monday and Wednesday. I do prepare normally a study guide, which I do think is already there for the exam. And study guide is where I pull out all the exam questions. So I encourage you to have a look of that. You remember, I don't offer answers or a key to the study guide in anywhere else than on the review sessions. So some students find it useful to watch that and work through it with me. Remember, it is an open book test that you can take from home. So you can use completed study guide. You can use your notes. All the questions in an exam are from the lecture video. So there's nothing outside of those. I think that I checked it. There is 50 questions. They might be multiple choice or matching or ordering or all kinds. At this moment, I don't think that I have there an extra credit question, but I might be busy during the weekend and come up with one, but I can't promise that. Maybe I will, maybe I won't. I'll let you know if there's going to be an extra credit question during the review session. I normally try to include those, but I don't always have those. If there is an extra credits question, I usually kind of give a hint during the review session as well, what it could be. So we'll see if I'm going to be able to come up with something. But that's next week, and it's available for the entire week. So you have entire week just to take the exam 3 and practical 3. But that's next week. The same goes for the practical. You remember that there is those review guides for practicals, and I really encourage you to use those practice versions of the practicals. There's good ones, a good one for the next week. Again, play through it multiple times. They are all fill in the blank questions, 50 questions in 60 minutes. Spelling is not something to stress. Mainly key terms. They're identifying organs or structures or specific parts of the structures. So it can be quite detailed. Again, open book. You can do it from home. You can use your notes. Some students prefer to take pictures of their practice versions of the practical, some write notes, whatever floats your boat, you can do that. But that's next week, like I said, available all week. What I want to do is to focus on this week. So this week, we have a lot of similar elements that you've seen before. We have the Learn Smart and we have the quiz. But what's new is that we are, instead of a lab, doing a critical thinking assignment. And critical thinking assignment is worth of 16 points, so it's quite a bit. So to give you a little bit of background, and some of you might have run into this already before, one of the things that college does, we do assess students' critical thinking abilities in all courses. So there's always some sort of a critical thinking element, and this one on the chapter about the fluids and electrolytes and pH balance is where we're looking at our critical thinking question on this course. So it carries quite a bit of points. I really encourage you to do that because it's not too hard of an assignment, but it requires you to do your own independent thinking. Again, remember, you can use your resources, and one of your resources is me. I am here to help you if you get stuck. Well, how do you do the critical thinking question? It's actually fully on Canvas, whereas the Learn Smart and quiz are with the McGraw-Hill. The critical thinking assignment is completed completely on the Canvas. And critical thinking question contains 2 case studies or scenarios, if you wish. And I do ask you to complete both of those. It's not that you get to choose which one you do. Each one is about 1 page, but do both. And I think they're ordered in a weird order, that first there's second and then there's one. How you access these is that you download within the Canvas from this assignment, the critical thinking assignment. It is a Word document, if I remember correctly. And then you upload a version of the document where you filled in your answers to the Canvas back. So I will go through those uploaded documents. If you don't want to upload your document, you can just simply type in your answers to the text box for the assignment submission. So you can either upload or type in your answers, but I want to see them in Canvas because that's where I do the grading. And you're asking me, well, what am I really looking then for? Well, there is actually what I'm looking for available for you as well. So the grading matrix that I will be using to assign the points for this critical thinking assignment is actually public. It's there on the Canvas. So use that to guide you. I would definitely, if I was a student, check what does he want me to find. So pardon me. So this is what comes up on Canvas when you click on the critical thinking assignment. It tells you a little bit about the critical thinking, what it's about, and then there's this link to download the document. And once you click on that, it downloads the document, which you will complete, and then you just re-upload that for me with your answers. Like I said, if you prefer just to type in your answers, you can type in them to the answer box as well. You don't have to worry about anything. I will be able to match them whose answer is what they get connected with your name when you submit, but it never hurts to add your name on that as well. But make sure you submit the version where you have your answers back to. This is what the grading matrix looks like. It really tells you step by step how much am I looking for how many points. So for example, here the maximum points were 4 points available. To get maximum points, you need to get 8 or 9 of the likely or unlikely questions correctly. So you can even get one wrong and still get the full points for that assignment. If you get 6 or 7, then you get little less points. If you get 4 or 5, you get little less points. And if you get fewer than 4, you get still a little point, 1 point. And if you don't have anything, then, well, then you might not get any points for that. Same as, for example, here, the other question shows that I'm asking you to identify either 8 or 9 relevant problems. That tells you when you're trying to find them, how many should I find? So I'm probably thinking that there should be 9 relevant problems that you can find. And then if you find less, you get less points for that. So that's how the grading is work. I would use that to guide me if I was a student. So that covers all that I had to say about the exam 2, practical 2, then about next week, exam and practical. And now we are ready to go through the chapter material of this week, which is all about fluid and electrolyte, fluid, electrolyte, and pH balance. This review doesn't take normally quite as long, so I'm expecting that it will go through quite nicely, but I do want to offer that review. The chapter is not quite as extensive, so we will jump out of this presentation, that particular presentation, so I will do so. I will open the correct presentation for our weekly chapter. And we'll use that for our review of the material. Let's get the right presentation open. Here, that looks more promising. So let's just share that with you. And I actually have quite nicely here just a couple of hints if you're wondering how to succeed in an exam. Really, the big thing is that be well rested, be well eat, have the time put aside, be comfortable. Since this is a take-home exam, take a quiet place where you can focus, where no one's going to come and interrupt you, especially with timed exams. That's important. Have your notes, have your materials around you. Have eaten, taken bathroom break, and all those. During the test, do really try to answer all of the questions. And if you have time, don't just finish it off early. Maybe use that time to review your previous answers. And of course, when you get the test back, so like for now, we did for exam 2 and lab practical 2, I really have provided quite a lot of feedback to you. So read that feedback, analyze where you went wrong, where you went right. Same as we revised just earlier on this session, the typical themes of the class, where students got most of the things right, where the common errors. Make a plan. How could you use that information in future to help you to succeed even better? But now that we've had a look of that, I think we're ready to move on and start looking at what are some of the important things that we need to know about the body fluids. We'll also review properties of water and water balance in the body. We'll spend a little bit of time looking at electrolytes as well as tonicity. And finally, we talk about pH regulation. So a lot of material, but really not that much in terms of the content. This is a lighter chapter in comparison to some of the things that we've seen before. So why is this important? Sometimes, for example, what you see when you do receive lab reports, you get these listings of various things that we can be checking on those. And some of those things do include various different values for materials that can be within the fluids. So really knowing what they are, what sort of amounts you should have, and where you should have them is important. And that's kind of what this chapter touches up on. There's typically some reference ranges, normal values, what we would expect to see. And if we're far off from that, that would give us an indication that it's something that we have to pay attention. So, for example, we've seen before the glomerular filtration rate, which was one good measure that we use to evaluate the functions of the kidneys, how well the kidneys are working. So that might give you an idea why we do have to learn about this. And really, one thing that I found is that a lot of the clinical decision making is based on these lab reports and lab results, especially in terms of the blood works, but also on other labs that we can. The other thing that kind of touches upon why this is important, we can have issues that we have too high or too low pH. So those would be some things that we could consider, and we kind of touch upon that at the end as well. But really, why all this matters? We do need to understand the basics of the fluids, electrolytes, and acid-base balance so we can read these lab tests and understand what's not normal. So we need to be able to understand first the normal so we can pick out what's abnormal. And in this week's critical thinking assignment, you actually get to do that. You get to pick out a lot of abnormal information from the case studies. So we want to know the normal first. Well, I take the discussion why we have it's the body fluids as the most important thing that we'll cover, and we start with that all the way back to your BIO181 or BIO156, depending on which general bio course you took. But even starting there, we talked about how so much of the earth is covered with water. And that went to say also that so much of all living organisms are made of water. So typically that value that you find in textbooks varies somewhere around 50, 60, 70%, depending on the publisher a little bit. And actually it turns out that it also is a little bit of different depending on the gender, or I should say sex of an individual, since gender is just a sociocultural construction, but sex is the biological one that we look in the biology. So typically we end up finding that males have a slightly more water content in their body, about 60% than females. If we look at the adults, and the difference is largely explained by the higher body fat percentage and also less skeletal muscle in the females than in the males. So these are averages. We also see that the infants, babies, have a high body water content. And this is, again, largely due to the things such as a low body fat percentage on the babies and a low bone mass. So more of their body in proportion contains water. In terms of the older people, well, they can have actually less water content in their body. So this is probably a slightly inappropriate comparison of the grapes and how we turn into raisins. We do actually literally dry up a little bit as we age. So what we should look at is where that water is located in the body. And we can do a division into two large places where the water is located, whether it's intracellular or extracellular. And we're going to start with the intracellular water concentration. So this refers to all of the water that's located inside the cells and sometimes abbreviated as ICF. The other value, the extracellular water content, refers to the water that's located outside the cells and is abbreviated as ECF. And you'll notice that about 40% of that 70, or about more than half of all that water that's in your body, 40% of your body is intracellular water. And then about 20% of your body is extracellular water. So most of the water in your body is inside the cells. And like I said, I would say that the proportions would be that about 2/3 of all water is inside the cells and 1/3 outside the cells. Where could we see then other things that we need to note? I kind of thought that one good thing to relay to you is why does the water concentration inside and outside the cells matter, is by talking about some of my previous own work that I did before I moved into teaching side of the thing. So I used to do medical research, medical imaging research, and especially focusing on cardiac MRI techniques. And even on that field that goes quite far, the basics, where the water is located, really matters. So what we see here is an MRI of a short axis view of the heart. We see left ventricle with its thick wall and a right ventricle next to it, a little thinner wall. The chest cavity, lungs above. I think we see nicely liver there. So these are the pictures that we would typically take with MRI using gadolinium to enhance certain tissues. And we see this bright white spot on the wall of the left ventricle there. Well, this bright white spot is actually indicative that there is some kind of a necrosis of a tissue. That tissue is not being perfused properly. So that tissue is dead. So my work focused on people who have had a heart attack or issues with the coronary arteries, and as a result, there's dead tissue. And my work focused on how much there is of a dead tissue on these patients and how much that tissue hasn't died yet. So we can kind of work out, is it worth doing a surgery, an invasive procedure, or is the death already happened everywhere that it will happen? Well, what I worked mainly on, and I'm not going to ask about this in an exam, I just kind of want to give you an explanation where the water is matters. What I worked on was novel sequences, new ways of looking at these things. And my imaging interest focused on T1 and T2 maps, which are not that different from what we see here on the left side. They look a little fancier because of the color that we use to signal the intensity of the signal. But what you see here is the same patient, same location. And you'll notice that on this T1 map, there's much larger area that's bright than on this traditional map that shows where the dead tissue is as a white area. Well, actually, this bright area is tissue that is slowly starving out of the supply of the blood. It's not yet all dead. It contains also some dead tissue, but not all of it is dead yet. Same as here on T2 image. So we see that there's significantly larger area that will die if we don't do anything than what's already dead. So on this patient, doing some sort of intervention would be worthwhile. We can actually salvage something. So that helps us to work through those. And actually what you're seeing here as this bright area is areas where there's elevated amount of extracellular fluid. When there is a heart attack, when there is a risk of tissue dying, the amount of water outside the cells increases in the cardiac muscle cell. So that's what I look, and what my interest was, the extracellular fluid in these patients and in the cardiac muscle tissue. So basic concept that we cover in very basic A&P classes that still plays an important role in kind of a far-going medical research. So what we're going to do next, we're going to go through the basic characteristics of water, and this should be all familiar from your BIO181, BIO156. So we'll go through them quickly, but I think that regardless, they're important things to notice. So let's start with the fact that water is important in transporting things. So, for example, in our body, most of the blood is made of water, and that's really the transport media, how we transport nutrients, energy, hormones, wastes, gases, oxygen, carbon dioxide, many things in the body. So water makes an excellent transport media. But it's not just only in humans, also in plants. Water is used to transport materials such as nutrients from the soil to the parts where the photosynthesis takes place, and then we can also get rid of some of the water through evaporation. The next one that I want to talk about is actually two concepts at the same time, cohesion and adhesion. So let's have a look of those. What we see with cohesion is when the water molecules end up sticking together, whereas adhesion is more of when water molecules stick to polar surfaces. So that would be two characteristics that we have for the water that play important role. So water really tends to stay together. We see it, for example, when you overfill a glass and there's this little dome above where the rim of the glass is. You can overfill the glass because those water molecules try not to start falling over. They try to stay together. So that would be a good example of cohesion. Adhesion, for example, what we see here, these water droplets attaching to the spider's web. Water does tend to attach to the to the other surfaces and polar surfaces. Same as if you have a glass of water, it usually tends, and it's not fully full, it usually tends to rise a little bit on the sides of the glass, and that's water attaching to the polar surface of the glass through adhesion. Then let's have a look at the surface tension. So water has a high surface tension. It doesn't tend to break. So if you are small enough, like for example, water bugs, they can use that and basically just walk on the water. Water also works as a great heat reservoir. So that's why seas and lakes, for example, absorb a lot of heat during the day and then slowly release it over the night. So temperature changes on the coastal area are lesser than somewhere like, say, for example, here in the Arizona desert, where it can get really cold at night and really hot at day, because there's really nothing binding that heat as similar to these large lakes or ocean fronts on the areas that are located on the coastal areas. Water, of course, have different forms, and this can become very useful for us. So the ones that we are mainly familiar are the ice, gas, and liquid. There is some other that we are discussing, but that's becoming more common, but we'll leave that outside from this discussion now. And then, of course, water makes an excellent solvent. So a lot of things can dissolve into water, and this makes it possible for us to transport materials with the water. So if we want something transported from one place to another, let's just dissolve it into the water then. So water carries all of these solutes, and in this way works as a great transport medium. Well, some do not dissolve in water, and examples of those would be, for example, the glucose, lipids, urea, creatines, and they are not really huge interest to us. The ones that we are mainly interested are the ones that actually dissolve in water, such as various salts, acids, bases, proteins, some of the proteins, and as they dissolve, they form charges, which you can see here. And these charges are going to be important in terms of how these materials behave. And that's what we're going to be paying attention, and what's going to be something that we want to notice when we discuss about these topics. There's some that are really important ones, and some that we find lots in the body fluids, for example, in blood or in between the cells or even inside the cells. So let's have a look of those. So what we have, the red bars show the blood, what we find a lot in blood. Blue bars, what we find a lot of in interstitial fluid, and yellowish ones, what we find inside the cells. So let's go through some of the most important ones. And the first one that I want to show are going to be our sodium and potassium. So we find a lot of sodium both in the blood and in the interstitial fluids, less so in the inside the cells, whereas potassium is found in great quantities inside the cells, not so much in the blood or interstitial fluid. Couple of other ones that I want to highlight. Here we see the chloride, for example, great numbers in the blood and interstitial fluid. We also see bicarbonate, some amounts. That was the carbon dioxide transport in the blood that we discussed in our previous chapters. And then we see various hydrogen derivatives as well, mainly in the intracellular fluids. So really, what do we take away from all this discussion? Well, what we take away is that water and electrolytes are connected. So when one moves, another one tends to move as well. So if we have water moving from one body compartment to another, the electrolytes with that tend to travel as well. So there's a connection between those. So they're interconnected. And I think that would be a good question in the exam. Are they connected or not? And here's some important sites where this exchange occurs. We're not going to spend too much time on that. I just want to show that this is something that really affects all parts of the body. So I guess that the next question is that how much should you drink in a day? About 2.5 liters sounds about right, of course, depending on the body size. So about 2,500 milliliters. So what ways do we get the water into your body? We'll have a look of that. But before we do, we also lose the same amount of water. So overall, this amount of water in your body is rather constant. So the intake and output of water tends to remain the equal. If you have one gaining or losing, then there's a net gain or net loss. But typically we try to keep that in balance. Well, the main way of getting water into your body is, of course, through drinking. So you drink or you eat wet food, and that's how you get about 60% of all the water that you get into your body. And that's driven by the sense of being thirsty. So when you get thirsty, that's your body telling you that you need more water in the system. Of course, like I said, moist food plays some role, and in many metabolic processes, you are also left with additional water, as we've seen. Well, how do you lose water? Here's the example of the metabolic processes, for example, cellular respiration, where we change sugars and oxygen into ATP energy, carbon dioxide, and water as a byproduct. But I was asking that how do we get rid of the water? Well, most of it is we get rid of it as urine. Just slightly less than 60%. There's a few other routes, some through evaporation, sweating, breathing. So there's different processes that play a role there. And of course, a little amount through feces. But it is really driven by the need to urinate, need to have a pee. And the concentration of the water balance in the body is really regulated by something known as osmoreceptor ADH mechanism. And we won't go into too much of a detail of that, but we will go through the water retention and water elimination by using a concept that we have seen already before. And when you talk about water balance and maintenance of water balance, many textbooks give you these complicated diagrams. What makes you thirsty? What's the water intake causes? Or little bit more simplified diagrams that are more of like a seesaw in their presentation. You can get pretty complex as well, but again, we see that there's two branches, too little or too much water. But we're going to make this much more simple. We're going back to something that's a very basic concept, and this is why I really wanted to talk about this concept at the beginning of our course. Concept of homeostasis. So homeostasis was all about maintaining consistent internal environment in your body. And we remember that we had this simple diagram that we could use to explain homeostasis. Well, we can use this simple diagram also to explain what happens when we have too much or too little fluids in the body. Let's have a look of that. We're going to start with the situation where there's too much water in our body. So you could think of our blood being too watery. That's the stimulus. That's the change. There's extra water in our body. Well, of course, we're going to sense that with structures called osmoreceptors, which are located in your hypothalamus of the central nervous system. And of course, the control center that we will see here is going to be the brain. We have the set point there, how watery blood should be. And if it's off from that, well, then we need to act. How could we act if you have too much water in your body? Well, it is from the pituitary. Pituitary releases the hormone known as ADH, and in this case, we're gonna release less of it. That's the key. So we're not gonna release as much ADH as we normally would. That results that we are actually excreting more water since we are not reabsorbing it at the distal convoluted tubule. And as a result, you end up peeing more. So you get rid of more water. This is how we deal with having too much water in the body. So you'll see this simple homeostatic loop mechanism that we learned can be used to explain many, many concepts, including this. What do we do if we have too much water in the body? We can look at an alternative situation where we have too little water in the body. Your blood is too salty, if you wish. So that's not a good situation as well. So again, we have osmoreceptors in your hypothalamus of the central nervous system. The brain is going to be the control center. There's the set point, what would be the normal amount of salts in the water. And if we need to act on that, this time pituitary gland from your central nervous system ends up releasing more ADH, antidiuretic hormone. More water is now taken back at the distal convoluted tubule. So as a result, we are producing less urine. So we're conserving as much of the water in your blood as possible. So I think that these two diagrams really show how we can use the basic concepts that we learn in a very efficient manner in our course material that is a little more advanced. I do want to talk at this point a little bit about the water balance in terms of the cells being hypotonic, isotonic, or hypertonic. So this is again something that I believe that you have covered earlier on, but let's have a look at those. The basic concept that we need to remember here is the concept that materials move from areas of high concentration to areas of low concentration passively. That's what happens normally in nature. In this case. So over time, materials move from that area of high concentration and spread out all throughout this space where we had this material. In this case, food coloring that spread out through the glass of water. So let's have a look at what sort of situations can we have. In case of a hypotonic cell, inside of the cell is more salty. So as a result, more water flows into the cell, trying to balance off that saltiness so that it be same on both sides. And when there's more water coming to the cell size grows bigger and bigger, and it might even eventually end up bursting. So how I think of the hypotonic cells is that I always try to make things that kind of remind me of something. And hypo sounds a little bit like a hippo. So hypotonic cells are these big swollen cells because water is flowing inside them. They're like hippos. The next one that I want to talk about is going to be isotonic cells. In isotonic cells, there's the same amount of salt both inside and outside of the cell. So there will be some movement of water in and out, but overall it's going to remain the same. There's no change in net gain or net loss. So how I remember isotonic cells, I think of these cells don't need to gain or lose water. They're perfect as they are. So these cells say that I'm so perfect, isotonic. And the last one that I want to talk about is hypertonic cells. In hypertonic cells, the outside of the cell is more salty than the inside. As a result, water now leaves the cell, trying to balance off this salt difference. And as water flows out of the cell ends up shrinking and might even end up drying up completely. So how I think of this, I think of a person who's very hyper. What are hyper people? Hyper people are very active. They run a lot. What happens if you run a lot and are very active? You become very skinny. So hyper cells are skinny cells in my mind. So really, again, use any ways that you can remember things to learn this. So just to summarize, in my world, hypotonic cells, let's go to the right direction. Hypotonic cells were these hippo cells, cells that were gaining water and then eventually growing bigger like hippos. Isotonic cells were perfect, isoperfect cells, don't need to gain or lose same concentration. And finally, hypertonic cells that are active, running a lot, becoming skinny cells. So how does this all relate to things that we need to know? Well, we want to make sure that as the waters move, well, we learned that with the water also the electrolytes will move. So if you go and exercise a lot and really sweat a lot, you will lose some water, but you will also lose electrolytes. So you might end up with a craving of salty things to replace some of those, replenish some of those electrolytes. I think a good example of that is when we are out and about in the summer in Arizona and we drink a lot. Just the drinking alone is not enough. You get this craving to replace some of the salts that you lost through sweating. And the question that I often ask from my students is that, can you die out of drinking too much water? And the answer is that it turns out you can. So what happened some years ago, there was a competition when the Wii game console was new. And I think that competition was a radio station competition where the theme was something like hold your Wii for a Wii. So they challenged participants to drink as much as they could in a short period of time without going to a bathroom so that they would get rid of that water. The idea was funny, playing with the words, but clearly these individuals had not studied anything about the tonicity. What happened then is that this individual, the cells ended up gaining too much water and eventually even bursting, and the individual died. So it is possible to drink too much water. That's why how we could have avoided this would have been, for example, by getting rid of the extra water, by urinating or taking diuretics or eating something salty to balance those salts, or drinking electrolyte-rich drinks and so on. So let's have a look at these electrolyte drinks a little bit. So when you exercise a lot, you might end up sweating, you drink water to replace some of that fluid, but we end up finding that the body fluids become too dilute and your brain tells you you need to pee some of that away. And again, you become dehydrated, although you just drank water. But how to deal with this is to drink something that contains electrolytes. So electrolytes are going to be important. Mainly what we want to replace are sodium and chloride, which we do end up losing a lot of in the sweat. And there are certain electrolytes that enhance the water uptake, such as the sodium chloride. And here's, just out of curiosity, I had a bit of a look. What do different kinds of electrolyte drinks contain and in how much quantities? Comparing that. So if you're interested, that might be a place to start that conversation or that consideration. And the last thing that I want to talk on our chapter material today is about the acids and bases. So let's jump there. You probably have seen the acid-base scale running from 0 to 14, 7 being neutral, any values above that being basic or alkaline, and values below that being acidic. Well, this is all a mathematical scale to show the amount of hydrogen ions and also hydroxide ions in the body fluids. So if you have acidic conditions, there's high concentration of hydrogen ions, and in basic conditions, we have low concentration of hydrogen ions. So why this is important is that pH really affects all of the functional proteins, biochemical reactions, and so on. So it needs to be regulated very closely within the body and kept within very narrow range. And what we see is that the normal blood pH, for example, is maintained between 7.35 and 7.45 of a pH. That's a very narrow range. And if we look after which you will die if you go outside that range is from 6.8 to 8 pH. Again, not that large of a range. Our body is, though, amazing at maintaining your blood pH within this very narrow normal range. If we have high pH, we talk about someone going into alkalosis, low pH, them going into acidosis. So you might have seen a lot of these things that are kind of popular and show up every now and then in diets, that eat an alkaline diet or drink this and that substance, and that will change your body pH. It's really not that they change your body pH. Your body pH will be always kept within this normal range. Your body is very good, very well adapted to that. How these diets might work is more of that the foods that happen to fall into one or another category are better for you, healthier for you. Not that they really are going to change your body pH. So that's a little bit of a hoo-ha and misconception that you might see if someone claims that. So how does your body keep this pH within such a narrow range? Well, there's three mechanisms that we're going to be looking at to regulate the hydrogen ion concentration in your body. They are buffers, respiration, and renal mechanism. And we're going to start with the buffers. This is the most rapid, fastest first line of defense of maintaining the body pH within the narrow range that we want. So buffers are chemicals that bind with either acidic or basic substances, and in this way getting the body pH where it needs to be. So quick chemical reaction. Respiration is also kind of not as fast, but still acting within the minutes, and in terms of managing and regulating the body pH. So there is a center at the brainstem for breathing regulation, and if there is a change in hydrogen concentration, we can adjust the rate and depth of breathing to be able to deal with that. And the slowest mechanism of all of these is, of course, the renal mechanism. So you end up peeing out hydrogen ions, and we just studied this in our chapter about the urinary system. So this takes hours, if not days, to act. And if we apply everything that we have learned, we can see that in case of acidosis, where we have low body pH, some of the things that we start to see is the nervous system becomes depressed, and you might get confusion, eventually even coma. This would be an example of this would be uncontrolled diabetes, the diabetic coma. So your body goes into acidosis. The opposite event, alkalosis, where the body pH is high, your nervous system starts to act a little bit hyper, you get this tingling sensation, and this might eventually lead into paralysis. So even the body pH, that has significant clinical relevance. And this brings me to wrap up this chapter by mentioning about fluid therapy, which is, of course, administering fluids to the veins. So they're intravenous fluids. We can't just put any water into there. It has to include electrolytes and salts in a proper concentration, so we don't upset the balance and we don't end up with those challenges that we saw with the hypo, iso, and hypertonicity. So really what these fluids that we use are salines in a medical use. They're pretty expensive salt water, but they're salt water or water that contains electrolytes and salts in an ideal concentration. And that brings us to the end of this chapter's new material. What I'm going to do now, I'm going to jump out of this presentation and we're going to go back to our original presentation to wrap up this session. And let's get that shared. Here we are again. And now that we have talked about the fluids, electrolytes, and pH and how to regulate the balance of those in the body, I just want to remind you I can facilitate if you want to retake any assignments, if there's something that you want to retry. I'm open to that. Message to me directly with details of specific assignment, or if you missed one. Usually the Canvas is the best way to message me. I have few messages there, but it was really important for me to make sure that you get your exam 2 and practical 2 grades back. So now that they're back, I can focus on those messages. Also remember that there is tutoring available. You don't have to have an appointment. That's all through the Cranium Cafe. And also I, as your instructor, will be happily taking time if you have any questions, either alone or as a group. I'll help you in any way that I can to excel on this course. And that brings us to the end of this session. So if you have any questions, anything that I can assist you with, all you have to do is let me know and I'll definitely help you. The Canvas is usually the best way to get hold of me. And a quick shout out that remember that this week we do have this critical thinking assignment, which we do want to complete. That's an important part of your coursework and also important part of assessment of this course that we want to focus on. So let me know if you run into any challenges. Make sure to complete all parts of the critical thinking, so both 2 scenarios. And that's it from me for today. Thank you for joining me on this recording.